Assalamu alaikum and welcome to this week's Journal Club, where I'll be presenting a paper published in the IAJ in 2026 by Tzanetakis and others. It is a systematic review titled Bacterial Diversity of Deep Carious Lesions, analyzed by next-generation sequencing approaches associated with different pulp diagnoses. So the objective of this study was to critically appraise and synthesize evidence on the bacterial diversity of deep or extremely deep carious lesions associated with varying vital pulp conditions using next-generation sequencing approaches. They also wanted to identify existing gaps in the literature and laid the groundwork for standardized, transparent future microbiome mapping research. So the sample size, they had 4 original research articles which met the strict eligibility criteria out of 1,900 initially identified records, and across the 4 included studies, the individual sample counts evaluated were 10, 75, 111, and 104 samples. For the methodology, they started with the database research. They did electronic searches which were conducted up to May 1st of 2025 in Medline via PubMed, Scopus, Cochrane Database for Systematic Reviews, and Web of Science. And the gray literature was searched via clinicaltrials.gov and OSF. And independent hand searches covered 4 major endodontic journals from 2010 to 2025, which included the IAJ JOE. For the eligibility, they included original English language clinical microbiology studies investigating deep and extremely deep caries, which was confirmed radiographically and clinically, and vital pulps with clearly defined preoperative pulp diagnoses utilizing the next generation sequencing. For quality assessment, the risk of bias was evaluated using a modified version of the Joanna Briggs Institute Clinical Appraisal Instrument for studies reporting prevalence data. And for the results, for the phylum level and for the genus and species level. For the final level, Tzanetakis was the most prevalent phylum across all pulp conditions, and its prevalence was significantly increased as pulpitis advanced to the irreversible stages, while actinobacteria, proteobacteria, and bacteroidetes decreased. And at the genus and species level, Lactobacillus was the most abundant genus overall, showing a massive increase in symptomatic irreversible pulpitis. And one study pinpointed that Lactobacillus reuteri as dominant in teeth exhibiting referred pain. Previtola and Streptococcus and Actinomyces also played notable roles. There are diversity shifts, which was that symptomatic irreversible pulpitis groups consistently demonstrated lower alpha diversity, which is microbial richness, but distinct scattered bacterial profiles such as beta diversity compared to normal or reversible pulpitis groups. And for the conclusion, this review, it maps out a preliminary but comprehensive blueprint of the deep caries microbiome across distinct pulpal diagnoses. And there is a microbial shift which occurs as asymptomatic cases transition into symptomatic ones, and this progression has clinical implications. It potentially helps dentists deciding between complete or selective caries removal technique. For the critique, this study has high methodological heterogeneity. The included studies lacked standardization, and they used different hypervariable regions, and they used disparate taxonomic databases. And also, it was inconsistent, it had inconsistent clinical criteria for diagnosing pulpal conditions. There was also a risk of bias concern because the overall risk of bias was rated as unclear for 3 studies and high for 1. So this is a major flaw which included poor description of sample frames and a lack of clear clinical metadata like age or sex. And thank you very much.